and it can undergo recombination. Whereas the mitochondrial DNA, during the fertilization, the spare head, it does not contain any mitochondria. The sperm head fuses with the ovum, and the ovum cell contains the mitochondria. As a result of this, you have the embryo. So whatever mitochondria is there in that fertilization, which through cell division will be transmitted and will present in all the cells, whether you are male or female. And that mitochondrial DNA is from the mother. So you could identify by looking at, we can trace back the mitochondrial DNA or look for the mitochondrial DNA at different levels from different people in different locations and use this as a tool to study the evolution. And that is how the classical paper came, indicating that all of us originated initially about 150,000 years ago from somewhere here, from somewhere in Africa. All of us, our grandmother is from somewhere in Africa, nearly 150 to 170,000 years ago. That is how they did the analysis, particularly from the mitochondrial DNA analysis, because it is essentially from the maternal side. Then one more thing, understanding how genes can be turned on or off. The genome analysis will tell us all these details about cell differentiation. So for instance, all the cells in our body have the same genome. Not all the cells carry out the same function. So we see through our eyes, not through our nose. And also we smell through our nose. We taste through the taste buds in our tongue. But whether you take the DNA from the tongue or the nose or from the retinal rods, all these DNA, the entire sequence is the same. But the genes, there are certain smell receptor proteins expressed in the nose epithelial cells. But these are not expressed in the retinal rods. The crystalline protein is expressed in the retinal cells. Rhodopsin is expressed in the retinal cells. Taste proteins, this is produced only in the epithelial cells of the tongue. That means there is a remarkable degree of differentiation, specialization, and this is through selective switching on or switching off of genes of these genomes. And this genome analysis is useful in looking at how these genes are expressed, how these genes are differentially expressed, what is the molecular basis for differentiation. Functional genomics approach, you go for the expression of the genes in a given cell and look for the proteins which are produced by these genes. Comparative genomics, that is, you have the data from different organisms and you can look at how these genes function and develop additional DNA resources. And now the work is going on in developmental biology, particularly with fruit fly Drosophila, so that we can use this as a model for studying developmental biology, how the eye develops, how the skull develops. These can be used as model systems, and more or less identical events are there with respect to expression of these genes during the development process. So by comparing these genomes from different organisms, it is possible. And now the genomes from a number of different organisms have been identified, sequenced, the simple fruit fly, mouse, C. elegans, yeast, rice, then you have the fish, the bacterial colony, and the human. And apart from this is facing a number of ethical and legal issues. So for instance, genome mapping or genetic profiling is it not cutting into the privacy and confidentiality of the genetic information? So for instance, is it advisable to tell you that when you do the genetic profiling, is it advisable to tell you that you have every chance of developing colon cancer? If this information is there, if it goes to an insurance company, if it goes to an employer, then certainly the insurance company will not be prepared to insure you. So the validity of that information, confidentiality of the information, the psychological impact, stigmatization, particularly in a society like ours, if there is a BRCA gene mutation in a girl and she is a marriage proposal, so it is going to be another situation. So look at the social implications of this new technology. Reproductive issues, including adequate and informed consent, particularly say for instance, you do a prenatal diagnosis and look at the possibility of having an affected child, then the genetic counselor will tell you that it is quite possible that you are going to have a deformed child. Do you want to abort that child? See, of course, the scientific observations and scientific data will have its own limitations. It is quite possible on one side that this data may not be 100% accurate, but with the available data, the counselor is advising you that there is a possibility of this child developing an abnormal condition. Do you want to have this pregnancy prolonged, or do you want to have this aborted? And that maternal instinct and other social obligations or social considerations will put you in a really psychological traumatic situation, whether to go on. So such issues are coming in. Then the clinical issues, including the educational doctors and other service providers. Many of the doctors, absolutely there is no science in this, the head of these clinicians. They don't know, they know only the manner in which the drug has to be prepared, they know the clinical symptoms, how the drug has to be prescribed, that is all. To use this modern technology, they must be adequately equipped. Uncertainties associated with gene tests for susceptibility to complex conditions. Fairness and access to advanced genomics. This may be available in other developed countries. It may not be easily available in developing countries. Access to the new information. It must be disposable. So even if there is a facility, unless you have the resources, you have the money, you may not be able to go for that sort of a test. So the access, availability, and other conceptual and philosophical implications regarding the human response, free will versus genetic determinism. As I said earlier, whether you want to have this child, it is your free will, whether to go for that or not. Whereas your parents, your friends will tell you that you don't have to continue with this sort of a thing. Why do you want to go for this? See, that sort of a situation. And also, of course, commercialization of these products. These technologies being used for making money. Just one. Another related thing is, of course, we must have thought of these things. News must have come in and out in newspapers regarding the fingerprinting, DNA fingerprinting. That is, as I indicated earlier, basically there are a large number of in this genome sequences. There are large amount of gem DNA, and within this gem DNA, there is a large amount of repeats. Larger repeats, smaller repeats, mini repeats, micro repeats. And these micro repeats are mini satellite or micro satellite DNA, some of the size of 10 to 50 and of the size of 50 to 100 DNA pieces. These are critically important. And this is a molecular signature. This is the molecular signature for each individual. So this has been used in UK. Alex Jeffrey developed the DNA fragmentation and restriction fragment linked polymorphism to identify the individual by fingerprinting. You know fingerprinting, what is meant by fingerprinting? You take the thumb impression, you have a characteristic profile of the finger. And this was something which was developed some 200 years ago by an Englishman, a revenue officer. He was a revenue officer. What happened was that he was in India. He was looking at the muster rolls. The muster rolls, at the end of the month, he paid the salary, paid the wages. He was not sure whether the same person was collecting the money again and again. So he said that, okay, we will have some marker. So he used the fingerprint. He took the fingerprint And then he followed this fingerprinting and sent this, look at that, this is a revenue officer, not a scientist. He sent this as a manuscript to Nature. And the Nature promptly returned the manuscript, rejected manuscript, saying that we don't have sufficient data. So what he did was, he waited for 25 years. He waited for 25 years, collected the fingerprint of the same set of people for 25 years, and then he sent back the paper demonstrating that during the 25-year period, this fingerprint remained the same. This has been extended to the DNA structure, and they identified restriction fragment in polymorphism. That is, fragment the DNA with restriction enzymes and use these fragments and take that as a molecular signature. And this is the profile of this. Number of cases have been sorted out, criminals have been identified, rape victims have been, the rape criminals have been identified. There are now one or two instances which has attracted attention all over the world. One such thing is that this is regarding the profile of a person who she married to an English person and was settled in UK. Their son went to Ghana, and when he was coming back from Ghana, the immigration people at Heathrow Airport did not permit him to enter. He was sent back. He was sent back, although he pleaded that he is the son of so and so. They did not take it. They said that no, you must be her sister's son. Her sister is staying in Ghana. You must be her sister's son. Then they fought. They approached Alex Jeffrey in Leicester. They got the DNA from his mother, his mother's sister, and his DNA, and the DNA from their other sons, and did the analysis. You see that these top all bands relate to his DNA, then his brother's DNA, and the other extreme, those relate to the DNA sample of his mother's sister's brothers, sons. So that showed that he could identify the true parentage, and then he was allowed to enter. Then of course there are other instances. For instance, you must be remembering that these American presidents always have a problem. You must be the news which came in recent years, Monica Lewinsky case. Not only Bill Clinton. Clinton's problem compared to what they had, the earlier presidents had. Just a simple problem. One of the former presidents of America, Thomas Jefferson. Thomas Jefferson was just after American independence in 1778. Thomas Jefferson was appointed as ambassador to France. At that time, he had made a slave as his mate, Sally Hemings. After the post-colonial period, Thomas Jefferson came back to USA. Later, he became the American president. But later, after about 200 years, in late 70s, the news started coming. There is a person with some name, Jefferson, who has the lineage is from Sally Hemings. The lineage, his grandmother was Sally Hemings. He claimed that he also would like to have a share of the ancestral property of Thomas Jefferson, the late American president, claiming that he is the son of Thomas Jefferson. This was a big case. Then they wanted officially, that is the thing, officially, Sally Hemings were only 3 daughters. So it was not possible, therefore, to go for a Y chromosome analysis. But still they did that. They did the Y chromosome analysis because Y chromosome is there. It is derived from the father. It is there only in the male offspring. They started analyzing the Y chromosome of Thomas Jefferson's official grandchildren and the grandson of Sally Hemings. Eventually they found that their Y chromosome fingerprinting matched. And the Jefferson became one of the legal heirs of the ancestral properties. So this is a much more serious problem than Bill Clinton recently faced. Similarly, there is another incident in Canada. There are 4 sons. It's an Indian family. They had 4 children. Father and mother living in India. Their sons are in India. They wanted to take their children. The immigration authorities ignored. On the pretext that probably, of course, Indians are clever in doing certain things. So they argued that they must be bringing their relatives or their sons of their relatives under this pretext. Because officially they have been living there for several years. Nobody knew that they had children, but they were keen. So they approached the immigration authorities, approached the blood sample of these people and their children. And they did the analysis. And their problem was, the people who did the analysis, they were in an embarrassing situation. Whether to reveal this information which they got to the immigration authorities, of course, they can release. They could not release the information to this Indian couple for the simple reason that out of 4 children, 3 matched very well. But the 4th did not match. See the embarrassment of the situation. They have been living happily, the husband and wife, they were living happily for several years. And all of a sudden, because they wanted to bring their children to Canada, they went for a DNA fingerprinting and they are ending up with this sort of situation. Look at the predicament of the scientist who did this analysis, whether to reveal this information. So this is the sort of technology that has come out. This is the sort of implications, social implications. This is the societal impact of this new technology, the genomics that is coming. Thank you for the extended information here.
I can see everybody who talks, I'll make them stand. Okay, please come back to the seat. Maybe you can take your tea with you. I hope you can hear me now. So I'm trying to get this slide back. We heard Dr. Sudhakar this morning, and the lecture was actually it's a very useful thing for me because I will show you a small clip which will make the students come back to the no connection. What is this? Maybe I hope. Human genome. Yes. Human genome. Yes. Human genome. Yes.
Containing a complete set of all our genes. The genome. Turn inside ourselves into a cell, we receive 23 pairs of chromosomes packed into a nucleus. Each chromosome contains a long DNA. If all the chromosomes were unwound, the DNA in just one of our cells would stretch 6 feet long. The DNA double helix contains 4 kinds of building blocks, and A always pairs with a T, a C, with a G. DNA contains information to make every part of our bodies with its 4-letter language. Each of our thousands of genes codes for a specific part. RNA polymerase copies the information in a gene into a messenger molecule. Messenger RNA The building blocks of messenger RNA and DNA are called bases. The bases on one strand of the DNA specify the order of bases on the new strand of messenger RNA. The DNA always stays inside the nucleus. The messenger RNA travels out into the cytoplasm. There, a protein-making machine called a ribosome can read messenger RNA into a particular protein. Every 3 bases of the messenger RNA molecule codes for an amino acid. Proteins are made of amino acids. 2 RNA molecules help translate the language of DNA and RNA into the language of proteins. 2 RNA molecules bring the right amino acids that the ribosome links together to make a protein. Proteins are the labelers. Some form structures like tendons and hair, others receive light, scents, and flavors, control chemical reactions, and carry messages between cells. To understand our molecular cells, scientists have read the 3 billion letters making up the DNA in the human genome. Different sets of genes interacting with complex environmental factors influence things like our looks, personalities, and risks for diseases like cancer and heart disease. A growing understanding of our genes and all they do will help us understand the complexity and the wonder of life. A simple friendship, a sunny day, our life.
Now you heard the Book of Life and saw the reality, Human Genome Project. And Dr. Sudhakar mentioned very clearly that the most important thing now is to see, to understand the expression of the genes. You have lots of genes, 30 to 40,000 genes inside your each cell. Once it gets differentiated, as you know, they take up different functions. You also mentioned that eyes, you go for seeing, you have smell, touch, feelings, all your pancreas for insulin production, brain for various functions which you will be hearing. Because I'll be talking about the brain and everybody has one, I hope, above their shoulders. Right? You have one. Many people they forget, they have a brain up there in the shoulders. Only when they have a headache, oh, they feel that there is something there. Okay? So you will hear something on the brain and which is very important. And we have to take care of that supercomputer very, very much so that your peripheral system also will be controlled. So each cell in our system, because we all come from a single cell, it multiplies, you get a ball of cells, then this ball of cells, it gets differentiated. And then the distribution of labor. To be distributed to various functions. Brain take up its function, liver take up its function, pancreas, skin. Now the task in 21st century for you, which I'll be concluding later, is to see, can you make the skin cells to function like a liver cell or a pancreas? The expression of difference, because the genes are there, can you erase the function, what it has, and reprogram it? Because we know where the genes are located. From the Human Genome Project, we know where the genes are located. The thing is that the functional, the expressional status can change. As you have seen in the Dola's case, I'm sure all of you know about the Dola's experiment, Wilbur has done, where the most important thing is not to make multiple copies of everybody. Somebody says, somebody wants to make 15 copies of their own, or Sachin Tendulkar, or anybody you want. That's not the main thing. As scientists, you should feel that the most important thing is that a differentiated cell, as he has taken the other cell, and it can be erased. The program it got can be erased and it can be reprogrammed. When you put it into egg cell, it should multiply and become an individual. If you can make that cell to put it in a pancreatic cell environment, it should function like a pancreatic environment, pancreatic cell. Can you do it? That's the biggest task you have in 21st century, which I will be concluding in my slide later. So, you have just started. I said there are so many start slides. You said there is a starting point. You have to see the expression. People have already started, so you take the relay stick and proceed ahead so that many of our system, which is not functional, can be revitalized and the function can be changed so that you can have a better, healthy, happy life, young life. Everybody wants to live young for long. Nobody wants to be old and live old for long. So live young, die young. That's what is the motto of aging, which I have done also for my PhD, aging. The motto is not to add life to age. No, age to life, but life to age. You have to add life to age. Everybody wants to live in the adulthood for longer time, collapse, then finish. Why somebody should have a burden? You should not be a burden for yourself and others. That's the most important thing in 21st century. So, otherwise, old folks home, many hospitals will be coming, which will be a burden for everybody, society and you and everybody. So, we have to take care of ourselves and see that we have a better life. So, the genomics, proteomics and human society, once you consider, you have to see how the genes expression can be changed. Once the expression is there, how can we make a protein functional, which is not functional? And so the alterations you have to do so that you have the appropriate functional molecule taking care of yourself for a better life. Maybe next slide, please. So I will be dealing with the brain. Never feel that brain, when you say that your brain cells I'll be taking, because I have taken my direction of research that way. Anybody can go in any direction. Somebody can go in plant, animals, bacterial system, anywhere. Nobody has restrictions. But essentially when it comes to the genomics aspect of proteomics aspect, it is same. Because genomics, when it comes, how can you play with the genes? And see that the functional, as he mentioned, you have the triplet codons. You have seen now the triplet codons getting the message for the specific amino acid to come for the protein synthesis. Can you, if there is a defect, can you change it? Or can you alter the change, the expressional status by altering the promoter function? Promoters control the function. There are so many factors from the primary messages, secondary messages, and you have tertiary, so many cascade of events. Finally, it goes to the DNA and alters, and the functional molecule is triggered to be synthesized. And cascade of events, as you have seen the central dogma, DNA, RNA, and protein, which is not existing now. You have a reverse transcriptase. But essentially, in our system, we have the message stored in the DNA, which is transcribed to the RNA, and that is carried, comes out from the DNA, nucleus to the cytoplasm, and then you have the protein machinery, the synthesizing machinery, the ribosome get attached, and the specific protein is synthesized. Then it is again processed. You have the post-translational modification. You have the post-transcriptional modification. So, at all these stages, it's all critical. We have to see that there can be a defect, and we can alter it if it is there. That's the most important thing. And now, I will say, the Center for Neuroscience. What we do there is to play with all these neurotransmitters. Serotonin, gamma-aminobutyric acid, acetylcholine, dopamine, glutamine, epinephrine. These are some of the major neurotransmitters which keep us to function appropriately. Once there is a disturbance in the balance of this, your life is disturbed. You are in trouble. You cannot function properly. You cannot, well, the normal activity is disturbed. So what we do is we measure this in different conditions. We go for various, these are all models for us. For example, pancreatic regeneration. You cut the pancreas, it will regenerate to a certain extent. You cut the liver, it will regenerate. You cut a small lobe of the liver, it will grow in 1 week's time. You cut 3/4 of it, it will grow in 21 days. The mass will be attained in 21 days. This is from our experimental evidence. Now we can see what are the changes occurring in these neurotransmitters. Always you should see that brain controls all our peripheral organs. And your activities, your talk, your vision, your smell, all hearing, everything is coordinated by the brain, directly and by indirect connection. Directly by the nervous system, and the moment you touch, or anybody touches, you feel it. There are so many, you know the network. We have a network of 3.2 million kilometers long. 3.2 million kilometers long network you have. So that the processing is so fast. So you know how complex and how efficient is your supercomputer. It's functioning. The moment somebody touches or you touch yourself, you feel it. There is a cascade of events which take place and the material is, you are visualizing, the function is taking place. In a normal, healthy individual, you have 3.2 million kilometers long network once it is properly used. If it is not properly used, it will get rusted. It will get rusted. So never let it not use it. If you don't use it, service and misuse. So always use your brain which is up above your shoulders. That's very important. If you don't use it, it will get rusted. Which I don't want to give you examples. It will take more time because you have to have your lunch, you will curse me. Okay. Now I said about this and we have the stem cells. I'm just briefly telling you of the things in general, later I'm getting to some specific things, finally concluding. Stem cells are the cells which are present even in adult individual which can differentiate to any cell. So you can take advantage of this. Now people have taken advantage of this and now they take even the cord blood. People companies in Bombay started collecting cord blood. The moment the baby is born, the umbilical cord is cut, you extract the blood and you keep the blood that has a lot of stem cells. You can store it. The baby, once he is or she is in 50 years, something is or 35, 40, there is a disease. You can make these cells to differentiate to the specific, locate it appropriately, see that the function is taking place. So it is 30,000 rupees they say now, and it is going to be a big business in the 21st century. Stem cells, maintenance, keeping it, store it for yourself, use it. So that there is no rejection, no immunological reaction will be there once you use your own cells. Because if you transplant something from others, there is going to be a rejection. Because there is a foreign particle coming and your system won't allow it, there will be antibodies and you end up in trouble. Then you have to use otherwise immunosuppressants. Once you use immunosuppressants, the problem is that you will be susceptible to any disease and you end up in trouble. So always if you can get your own cells to function, either you make your cells to function the other way or you keep your cells, store it, which you will see as a beautiful slide later. And so stem cells research in 21st century is very important for the young scientists coming. You have to see that that's of so much importance for you. Alcoholism is another project we have where we want to see how can you make a person who is alcoholic, not alcoholic. The only thing is that you make him, the moment he takes alcohol, he should collapse. He should faint or he should be allergic. Then you will be scared to take it. So there is an enzyme we are working on, aldehyde dehydrogenase. Because the moment somebody takes alcohol, it will be converted to first aldehyde by alcohol dehydrogenase. Then aldehyde dehydrogenase to acetic acid. Then it's excreted. When you consume it daily, your efficiency increases because the alcohol dehydrogenases and the other enzymes get stimulated, they will degrade it fast. So you can consume more. Now the thing is that there are few the Japanese and Chinese trade with human beings, they have this enzyme aldehyde dehydrogenase not there. They don't have the expression of aldehyde dehydrogenase. So what happens is, the moment they consume, they become allergic, even they faint, and they should be fatal sometimes. So that is taken advantage, and people make tablets which can inhibit aldehyde dehydrogenase. It is given in the market now. But the thing is that the person should be careful to give the tablet. If the husband is given more, he will be no more. It happened 6 months ago, or I think a year ago. See, one tablet was given to inhibit, and the doctor specifically told, don't give more than one. She got tired of it and she gave 2 tablets one day. He went and drank so much, he collapsed, and he never woke up. That's it, end of his life. So we have, once it is in our system, we have the regulated system. We can regulate it. So we want to see, can we make it regulated in our own system? The gene regulation with the factors. Another thing is bone healing. For years, when I had collaborated program with Cochin Medical College Orthopedics Department, Dr. P.S. John, who is head, for years they know that if you have a peripheral fracture, If you have a leg fracture or a hand fracture, suppose if it heals maybe in 45 days, once you have peripheral fracture and head injury, the healing is fast. We take maybe only 30-35 days or 30 days, much less. But we cannot break somebody's head whose leg is fractured, so they will kill you. So the only thing is that we find the fractures which are important, which is additional. So when there is a head injury, the supercomputer or the powerhouse, there are so many factors released. These neurotransmitters are triggered. They influence in the healing process, and we have experimentally proved. And it's already, it has come in paper, it has gone, we have published also some of the things in it. So and even some of the doctors, they got gold medal when they presented in conferences. They are so happy. And we see that you apply some of these neurotransmitters in appropriate concentration, appropriate combination, the healing process is enhanced. So right now we have to go to the molecular mechanism, how we can proceed with it, and how effectively we can do it, which we are in progress. Another thing is herbal medicine, which you all know, you eat every day so much spices, so many medicinal plants, spices, many of the things which you consume, they all have medicinal effects. And we have lots of these medicinal plants, and especially I can say the Idiki district should have so much, because the Idiki area, you have so many, even the lychee, or you say pepper, they all have effects. And some other medicinal plants, kolam, and so many other things, they all can have, even our curcumin, what's that, manjal. You get, it has got so many effects. It has got anti-cancer effect, anti-diabetic effect, anti-ulcer effect, lots of. So which you consume every day, you put in your curries. And so, curcumin is very important. Otherwise, the same way, there are so many compounds present in all these plants, medicinal plants, Ayurveda, many of these things they put. Even Nagarjuna is very close to you. I had visited Nagarjuna once. They make hundreds of combinations and various anti-ulcer property, anti-diabetic, anti-cancer property. Many of these things which gives, there is amrita or something is there, and which, one more thing is there, I think, which is used for memory. Everybody says they want to improve their memory too much. Vermi is there. So there are so many plants which are present here where the glycosides, polyphenolic compounds and alkaloids present in different combinations can have very good effect for our healthy life or happy life. Once you are devoid of the disease, you end up you are happy. Next slide please. So anyway, some portion of it is cut. Biotechnology is the term for group of technologies that can burst you understand. See, the main thing is that can you make the information you have from the biological system to an industrial application? Can you make, even for example, medicinal plants, get some of the secondary metabolites, use it for your benefit, and you can have industrial scaling up. Can you scale it up? You have it in the laboratory, then you scale it up and see that it is put to the market for people. It can go as food supplements, or it can go as medicines, which some of our companies are working, and you have tremendous opportunity there. And you are the treasure here, we have lots of, but we have to use it. How you use it, and you plan it up and proceed ahead to the industrial level. Next slide please. So let's say again, just to show the brain, different regions. Even when you speak, or when you sing a song, or when somebody is a talent, they have a specific talent, the specific region of the brain is activated. That region is activated, now we can visualize that specific region. The nuclei can be visualized by functional MRI, magnetic resonance imaging, functional MRI, can even visualize which region is triggered. The moment that is defective, that cannot be seen. So, there are so many ways we can visualize where the function is taking place. Now the thing is that can you modify it? If there is a defect, can you correct it? Because brain cells, neurons, so far we cannot make it to divide. Maybe after first 6 months to 1 year, even there may be few neurons dividing. After that there is only death. It's the same way for muscle. Muscle is also a post-mitotic tissue. Liver, pancreas, they are pre-mitotic tissue. brain muscle, they are post-mitotic tissue. They cannot divide. Only they maintain the structure. Now the thing is that in brain you have supporting cells like glial cells which can link it. So you can see old people, they can't coordinate memory. They forget what they have taken in the morning, whether they have taken breakfast. But they may be remembering things 50 years ago what happened. The immediate past they forget because the link, the message transfer is blocked. Now we have to see that the link is established if they have to be cured. Or if they, so there's a deterioration, it's a slow process, but once it comes to a stage, then reversal is difficult. So we have to see that how we can prevent it first, then go for a slow cure. So you have the task to see or give image message to the people how they can protect their cells, which are very important for their life. They should not damage it. Because accumulation of the damaged things is not diabetes or any brain disease, is not one day. Even from the childhood, if you maintain your diet, your exercise, your thinking and your lifestyle is maintained, you can prolong your disease coming. But you will think about it, postpone all the problems, you postpone all this modification of lifestyle till you are declared that you are a diseased person. Because that is a critical stage. From the 100% cells, if damage is finally when it comes to maybe 40% you are alive, you are in trouble. So protect yourself maximum. We have so much of the environmental toxins and many things, but there is a repair mechanism which is happening in your system. So see that your repair mechanism is effective and see that it is functional and you should not deteriorate too long. Because the moment your repair system is defective and you are damaging or damage is more, what happens is kill the cells die, tissues die, organs die, finally you die. So try not to kill the cells and so that you can protect yourself in the healthy area. Next slide please. So these are the neurotransmitters we play. Chemical messengers released by neurons. The same thing. So we play. The balance of this disturbed, they play abnormally. Everybody plays abnormally. So better keep your balance, things appropriate, at least to reasonably alright condition. Next slide please. So let's say message transmission. See, it goes from here and you have the axons and the message is released here, the synapse, and you have the receptors receiving it. And the appropriate receptors take up the message, you have the second messengers, you have cascade of events take place, finally reaching to the nucleus and the DNA on the specific region, the gene. The gene is the piece of or portion of the DNA which can functionally go for a specific molecule, functional molecule like protein. So the message is transmitted so fast from neuron to neuron and then to the peripheral tissues because there is connection from the central to the peripheral. Next slide. So you can see the central connection, neuroendocrine regulation. If you take the brain can have a direct innervation to liver, pancreas, and you have an indirect system coming to thyroid, adrenal, and which can go to the peripheral tissue. So there is always a direct and an indirect connection. Both are important. And never think that one can be substituted for the other. No. Both together functions and maintain our day-to-day life. So each system is controlled from here. The hormonal pathway is the direct one, and you have the direct pathway which is connecting, as I told, the network, connecting to the various peripheral organs. And each cell, the message reaches. Next slide please. So you can see, even people say diabetic neuropathy, you have brain altered neurotransmitters, the receptors, increased sympathetic stimulation, pancreas, altered neurotransmitters and the receptors, beta cell death, decreased insulin secretion, which end up in high glucose level or hyperglycemia. Low glucose uptake by cells, liver, brain and others, means you have, even though you have hyperglycemia in the circulation, high glucose, you have hypoglycemia in cells because the glucose is not gone inside, it's not pushed in there itself for its function. I can see some of them talking and they are talking, see, I can see, see this people are talking there. There is one, forget about it. So, hypoglycemia, it says, and you see the brain cell damage, finally diabetic neuropathy. She didn't notice it. She's still there with her scarf. She was talking. Don't disturb others. Okay, next slide please. So even when I say a simple neurotransmitter or a vitamin is very critical, which you can see all of you now heard about vitamin B6. I'm sure in your lifetime you have taken once a vitamin. If you have not taken, you are taking through your vegetables and fruits. Okay? So nobody can say that you have not taken vitamin B6. But vitamin B6, we have done experiments, we have about 15 papers related to this. When I was in Canada, we have done. You can have a B6 deficiency, which is again going to decrease the serotonin because there is a decarboxylase enzyme in the synthesis of serotonin, GABA, dopamine, serotonin. So once you have a deficiency of this, there is a decreased serotonin and GABA function. Now that can lead to hypothyroidism, decreased thyroid function. Hypothyroidism, that means decreased thyroid function. Once there is decreased hypothyroidism, can lead to hypertension. Because we have done experimental evidence. So we have experimental evidence. We used to get 10 pregnant rats. The gestation period for rats is 3 weeks. We used to get 2 weeks pregnant rats. You start feeding the vitamin deficient diet to the control, you add only 100 milligrams of this vitamin to the total 1 kilogram diet. The other one you don't add that 100 milligrams diet. The pups, the babies born will be abnormal. That 1 week's time, it takes a day or two to get into the circulation deficiency. The babies born will be abnormal and they die in maybe maximum they survive for 1 week or 2 weeks. If you continue feeding this vitamin deficient diet. Now if you feed that 1 week after delivery, can go up to 4 to 6 weeks. Even in the adult, we have seen, you feed this deficient right to adult rat, you have a central nervous system controlled hypertension in 8 weeks via thyroid function. So that can lead to, once you have hypertension means you have this hormones, epinephrine or norepinephrine high, which is the indication that you are hypertensive, which have to be reduced. So vitamin B6 can suppress the centrally mediated hypertension. You don't require it, but so much. Very little is enough. Now, another thing, we used to get hospital samples from babies, epilepsy samples, sometimes where there is B6 deficiency, they go to coma. Some cases we can treat with high dose of vitamin B6. Whereas some cases we cannot, because vitamin B6, once you take it, it has to be converted to its active form, pyridoxal phosphate, by pyridoxal kinase. Now once the kinase enzyme is not active, when this B6 activity function is not properly carried out. So such cases we have to modify the enzyme and see that the active form of the cofactor is produced. Otherwise, it cannot begin to the function. So you can see how critical is a small vitamin. I just give you one vitamin. So we have lots of other vitamins which are very critical for our day-to-day function. Next slide, please. So once you have taken monoamines, NAD, FAD are increased significantly in diabetes. Just now you have seen up to hypertension. Once you are hypertensive, the norepinephrine, epinephrine you have high, which will inhibit insulin. So you have decreased insulin and you become diabetic. You are declared as diabetic because it's a progressive one. So once you have more of this norepinephrine, epinephrine, it will be looking at cellular diet and it's a gradual deterioration of the function. Now, platelet monoamines are also decreased. Platelets itself is less in diabetic patients. As you know, in coagulation, always you hear that diabetic patients, the wound healing is not proper. You can hear later very severe cases, legs getting amputated. They don't have any sensation in the peripheral leg or hand, leg most of the time. and they cannot function. Their nerves degenerate and their function is not taking place. Wound healing is not proper. Why? We can experimentally prove because the moment there is a cut, the wound is coming. I think I should know what time I should stop. It is 1:15. Dr. Stephen, the organizing secretary, is moving around. What time I should stop? 1:30. 1:30. I'll stop at 1:30. Okay? Because I got only, see, it's not. They took the time. Maybe you took the time. Okay. I can blame only the organizing secretary. Okay? Nobody else. Anyway, the thing is that once you have a moment there is a wound, the wound comes, initial cut is formed by the platelets. And then the other cells occupy and you have the vital network coming. So when the platelets are less, your initial wound blockage is less. And you have high circulating glucose in a diabetic patient. Very nutritious state, nutritious place for the microorganisms to grow. So they grow and the infection, so the healing is not proper. Okay, so we have seen platelet formations are increased significantly in diabetes, amyloidosis, increase in monoamides in plasma and proportional reduction in platelet counts. Those are the platelets less and glucose high, which is very good place for the microorganisms to grow. So the wound healing is not proper. We have experimental evidence. Next slide, please. Second, just to show the experimental thing. In difference, there is a difference in young and old also. You can see monoamines and metabolites in striatum, young and old rats. As you grow old, these functional molecules are different. So you have to balance your system to maintain it appropriately. You can see dopamine goes up. It's up in young, old it is down. Because some of these are very important for behavioral, very important for our behavioral thing. We have the behavior is controlled by some of these neurotransmitters. They are balanced in different regions. So it's very critical. Next slide, please. So some of the diseases, central nervous diseases like Alzheimer's, you would have heard, as I mentioned earlier, people forget when they get old because the link is not set. So they can't remember the immediate past, but they can remember the old ones. So try not to damage your nerve cells. Parkinson's disease, you have heard. Mohammed Ali, boxer, he has Parkinson's. There's a continuous pass, it stops because it is the function is not continuous. There's interrupted functions so that they cannot function normally. Then you have depression. Everybody would have faced depression during exam failure, somebody cheating, all sorts of things. There will be depression. Everyone would have. But the most important is how fast you can recover from the depression. If you accumulate your depression, go on adding, you end up in trouble. Finally, you have to go to the hospitals. You have to put a pipe very close here. Or there are so many coming up now where you have to go there, the remedial measure. The earliest you go recover, the better it is for you to function normal. So, depression, it's a very common one, but you have to be careful. It's also a disease which can lead to various problems. Then you have other brain disorders which I don't want to mention too much, which you have Huntington's disease. There are so many other diseases. Peripheral system, which I just mentioned to you, hypothyroidism, hypertension, diabetes, they are all linked. The moment there is a deficiency B6, you have hypothyroidism, which can lead to hypertension, which can lead to again diabetes. So all connected. Next slide, please. The adrenal hormones and adrenergic receptors have an important role in glucose homeostasis and nerve function. This is the adrenal gland just above the kidney. Okay, next slide, please. Parkinson's disease. Monoamine transmission or dopamine transmission is inhibited in the substantia nigra. Basically, amyloid corpus right of the brain. So you can see each region has its own function. Substantia nigra, when you say, it is very important for the dopamine in the corpus striatum. The synthetic body is in the substantia nigra, the end is in the striatum. We can experimentally do even damage one side of the substantia nigra. The other side will be taking over. You can do an animal. The animal will be rotating like this. If you damage this, the animal will be continuously rotating like this. Even it is the same case with humans. You damage, the person will be continuously running around. And you can see they rotate and rotate and rotate. You cannot stop. Because then see the balance is very critical. Next slide, please. Norepinephrine, dopamine, the pancreas is a function of age. even as a function of age, norepinephrine can increase significantly in diabetic old rats, while it is unaltered in rats. Dopamine content decreased as a function of age in control rats, while it is unaltered in diabetic rats. So it says again, there is a functional difference as you grow old. As I mentioned, try not to damage your system. There is a progressive damage and also a repair. So try to see that your damage is minimized and repair is enhanced. That's to maintain your better life. Next slide please. So you can see differentiation, growth, maturity and senescence occurs due to sequential activation and repression of genes. You walk through the life. So see that your appropriate genes are maintained during the adulthood, and so that the good maturity is maintained for long and senescence is blocked to till you want. Because some of the young kids, when I talk, they say that some of them they want to live for 500 years. Sometimes they don't want to die at all. From the schools, when we talk to them, how long do you want to live? 500 years. They say, I don't want to die at all. So they had to maintain it. Next slide please. So again, this is a procedure how you do a receptor binding expression process to show that the experiments how they are done. This again how you can do a receptor gene expression. How it is expressed. As you do an enzyme activity, a kinetics, KAYAL and Vmax. The same way Vmax, receptors also we can do, which will be an index to measure the expressional status. Next slide please. So that is again showing Vmax and a KD, which shows the number of receptors and the affinity. As we have Vmax in the enzyme, K, the affinity for the enzyme. We have receptors also. The number of receptors located. Any molecule released, the receptors have to be there to receive the message. Next slide please. Again we have done genus special studies, we can do in another way, PCR. You will see some diagrams. PCR is polymerase chain reaction. RT-PCR is reverse transcriptase polymerase chain reaction. You have another RT, real-time PCR. As it progresses, you can visualize. You will see the data now. So Dr. Sudhakar mentioned about DNA analysis and all this. If you get a little bit of the DNA, even you can amplify it. So people take advantage of it. Even reverse transcriptase, the enzyme which breaks the central dogma, DNA, RNA, protein, making RNA to DNA, cDNA, complementary DNA, is used here for the any mRNA you want to see that the expressional status of this specific protein. You can isolate the mRNA, get the cDNA, amplify it, measure it. Then you can see how much the genus is expressed. Next slide please. So again, just to show a illustration of, for example, 5ST1 here receptor which is done in our lab, one of the PhD students. So any condition, you can see an sham operation, 72 hours, pancreatectomized, 7 days. 3 different conditions you have. That's the control. There's another one. There's 72 hours pancreatectomy you do and wait for 72 hours. Here you wait 7 days. So you can, there is an expressional status difference and it is reversed. So you can see there is a change we can observe and which is reversed after this period. Which again saying that can you make a molecule to a controlled way? So during a disease condition, this deteriorates and then it reverses. Next slide please. This is the real-time PCR thing. See, when you do a PCR or RT-PCR takes 2 days. Because you have to do a PCR amplification. We have to run a gel, observe it, the pictures. In the real-time PCR we can visualize the progress and we have a crossing threshold. Because you keep, because initial changes 1 to 2, 2 to 4, 4 to 8, like that multiplication goes about 30 cycles. So you can put the cycles around 10 cycles or 15 cycles as a threshold. Then you can see the crossing. The same things. Sham, 72 hours pancreatectomy, 7 days. You can see the crossing threshold difference. That indicates there is a difference. You can see here, I think, what is it? The blue. The blue one is the sham and the 72 hours, the grey one. And you can see the third one, it is coming back. So, an experiment you do after this period is reversing, which you can visualize it. And the standardization. No need of a gel, can visualize at the same time, can do. So So sophisticated instruments are coming for you in 21st century to do a lot of things same time. Next slide please. So you can, about the plant, I have a few more minutes. Plant extracts, you can have principal components. They can be as a neurotransmitter modulator. It restore the altered brain and peripheral neurotransmitter functions observed in various diseases, which can be a therapeutic tool. So you have ample of opportunities to pick up a plant, modify its combinations, do it. And a representative company is very close to you. Next slide please. So an example is Poulose as a potent anti-diabetic drug. It regenerates the damaged pancreas, increases the lower platelet count, decreases the elevated level of epinephrine norepinephrine from the brain, decreases the increased acetylcholinesterase activity. These are the few parameters we have looked, and it's an example to be given. Poulose can help us or help a patient, diabetic patient, to improve its disease condition. Next slide please. Dr. Sudhakar mentioned about the ethics and the advantage and disadvantage. See now, fetal disease and diagnosis and management is becoming, some places has become a fashion to people see whether it's a boy or girl. Actually, it should not be checked. We can see in the first 2-3 weeks even, you can get the placental fluid. You can see the cells there, analyze the DNA, you can see whether there is the baby is XY, XX. Whether the baby has 23 pairs of chromosomes or an additional one. So, but the technology has improved so much, we can analyze it even from very early stage. But it should not be misused. The moment you take the placental fluid, it is definitely going to make some problem to the baby. Even the ultrasound scan is going to create a problem to the baby. So, it should be a minimal use of ultrasound or minimal. When this is a suspected case, you can go. That will be advantageous. Otherwise, nobody should go for it. Because it is going to affect the baby over there. Everybody is going to go through the scanning now every month or every visit. Scanning. It's not good. Which should be affecting because the ultrasound waves are going. It is going to affect the sensory organs. The baby will be affected not initially, maybe after 40 years or 50 years. Because if you kill 10 cells, the after effect is going to be seen after years. So only suspected case, when it is really important, then such things should be gone. Then you can go and centrifuge fluid. You can get the sex determination, biochemical enzyme studies, you can karyotype it, see the number of chromosomes, everything from the initial first month even you can see it. It's possible, but it should not be a routine check. Never. There is ethics going on. And it should be people, those who are scientifically knowing things, they should be delivering the message to the people. Next slide please. As I mentioned earlier, since the stem cells you have differentiation. It can differentiate you. You have the blood cells, the nerve cells, the muscle cells. So, which is going to be also another important area of work in 21st century. Even brain has stem cells. Your bone marrow has so much stem cells. And you can make the function. You can store the cells as mentioned. But everything has its own bad effect, good effect. You have to balance it. Next slide please. So in future, commercial development of biologically active new medicinal products and plant origin, we bought and regulating blood glucose level, regeneration of pancreas, prevention of tumors, and so medicinal plants have tremendous importance in the coming century, and we have rich resources in our country, in our own states, specifically in this area also. We have to make use of it. Next slide please. I think that's the last slide I'm going to show, then I will get questions if anybody wants. See, this is what I was telling. Can be a gene bank. There can be a tissue bank. See, you can give a gene laboratory. Give any tissue and reprogram it as per your reality in 21st century. People lined up. Any cells you can spare. It can be skin cells. You can spare some of your skin cells. Ask them to make pancreas, liver, neuron, kidney cells, whatever you want. Otherwise, next alternative is that make this much region of your skin cells to function like anything you want. You can ask them. So it depends. The gene laboratory whether you can give it and get it done, or you can make it in your own system. And that's your task in 20%. So the expression and suppression in a programmed way, if you can make it, you can make your life much, much better. You can have a young, healthy life for long. Thank you.